Hello everyone. In this video, our SNSCAHS students of the Department of Cardiac Technology, Operation Theatre Anesthesia Technology and Cardiopulmonary Perfusion Care Technology students are very much eager to share their biochemistry important topic enzyme inhibition. So they are very much eagerly waiting to take the enzyme inhibition concept into the role play and the real life case scenario. So let's dive into the video and I welcome all the students of the allied to role play their enzyme inhibition topic.
Hello everyone, today we are going to learn an important concept in biochemistry, that is enzyme inhibition. Enzymes are biological catalysts that speed up the chemical reactions. They have a specific region called active site where the substrate binds and get converted into product.
Enzyme inhibition is a process which a molecule called the inhibitor binds to enzymes. It decreases its activity. Now let us understand this concept through a role play.
Normal enzyme action is when the enzyme is binds to the substrate, it forms enzyme substrate complex and releases products.
I am the enzyme amylase. I have an active site specific for my substrate. I convert starch into simple sugars. I am the substrate starch. I bind specifically to amylase.
Amylase, starch, they both combine and they give enzyme substrate complex.
I am the product of simple sugar formed from starch with the help of amylase.
This is a normal enzyme reaction. Now let us see how the inhibitors affect the process.
There are three types of enzyme inhibitors are here. I make competitive inhibitors, non-competitive inhibitors and competitive inhibitors. I will explain about competitive inhibitors. Competitive inhibition occurs when the inhibitor resembles the substrate and combine with it for binding at the active site of the enzyme.
I am succinate dehydrogenase. I will convert succinate into fume. I am succinate, the nature substrate. I will bind to the enzyme succinate dehydrogenation.
I am beta nanoate. I resemble succinate, so I will compete for the active site.
Here, nanoate acts as a competitive inhibitor and blocks the active site.
Non-competitive inhibition occurs when the inhibitor binds to the site other than the active site, called allocytic sites, and changes the shape of the enzyme.
I am cytochrome oxidase enzyme and help in ATP production. I am the substrate oxygen I required for energy production.
I am inhibitor and cyanide. I bind at a different site of cytochrome oxidase enzyme.
Cyanide inhibits cytochrome oxidase and preventing ATP production. This leads to be energy failure, hypoxia, anoxia, and can be death.
Uncompetitive inhibitions occurs when the inhibitors binds only to the enzyme substrate complex, forming an inactive complex.
I am lactate dehydrogenase. I convert pyruvate into lactate.
I am substrate pyruvate along with NADH. I bind with lactate dehydrogenase enzyme.
I am inhibitor oxalate. I bind only to enzyme substrate complex.
Oxalate inhibits lactate dehydrogenase forming an inactive complex.
Irreversible inhibition occurs when the inhibitor binds to the enzyme permanently. This usually occurs through strong covalent bonds and this makes the enzyme inactive.
I am acetylcholinesterase. I break down acetylcholine in nerve transmission. I am substrate acetylcholine. I am essential for nerve supply. I am inhibitor dipropyl fluorophosphate. I permanently inactivate the enzyme. This irreversible inhibition leads to accumulation of acetylcholine and affect the nerve function.
Our students of CT, Simplicity and Motility have clearly explained about the enzyme inhibition as a role play. So our students have involved actively in role role play of enzyme, substrate and